Perfect timing. We had a few, yes, perfect timing. We had a few issues at the beginning, but we are right on time. So hello, everyone. Welcome to the second seminar of the series, AEESP, AJAR Future Faculty Seminar Series. Today we have two outstanding speakers. First, we're going to have Dr. Kiranmayi Mangalgiri, and then we're going to have Dr. Elyse Stachler. And at the end, please remember that we're going to have 15 minutes after the talks that is going to be for informal networking, so we can talk and you can ask all the questions that you may have about the program or something else. And after each talk, we have a series of questions. When you ask questions, please introduce yourself because we want to know who you are and what you're doing. And without anything else, we're going to just get started. Our first speaker, as I was saying, is Dr. Makarkiri. She is currently a Chancellor's Postdoctoral Fellow in Chemical and Environmental Engineering at the UC Riverside, working in UV-based advanced oxidation processes to treat brine products of RO. And she did her PhD from the University of Maryland, Baltimore County, working on photolytic fate of antibiotic active contaminants. And she has received numerous awards for her research and outreach activities. And she's going to tell us all about this and her work in this presentation. So Kiran, once you're ready, you can start. Thank you.
Excellent. Thank you so much, Lucia, for that introduction. I'm really happy to be here to be presenting some of my work with you all. And I want to thank AEESP's AJAR committee for giving me this platform. This is a very exciting opportunity for anyone who is on the job market. And so I really appreciate this opportunity. So I chose the title for today's talk, Let There Be Light, because it's also the motto of the University of California system. And I thought it would be very appropriate for a talk that's mainly going to talk about photodegradation of contaminants in water and wastewater treatment. And so before I'd like to actually thank the University of California presidential and all of my advisors, mentors, the graduate students and undergraduate students who worked with me in doing all of this research. So, so before I actually jump into some of my data, I wanted to take a quick minute and just recognize how far we've come in our understanding of what is safe drinking water. So the earliest regulations in this country for drinking water only looked at bacterial counts and metals and came into effect somewhere around 1925, so around 100 years ago. And before that, potable water or drinking water was just meant to be having a pleasant taste or smell and color, right? And it was just called pure and wholesome. And it's only in 19
Hey, Kiran. Maybe we can ask her to turn off her video.
Yeah.
This is lesson for everyone. Don't use Wi-Fi when you're on an interview. Cable.
Kiran.
Past 1995.
Kiran, can you stop for a second?
Sorry.
You keep cutting out. Can you just not use your video?
Oh.
No, she's gone.
Yes. So this is a good practice. This is a good practice for all of us.
All right. I'm back.
Kiran, maybe you can not use your video. Can you hear me?
Yeah, I'll turn off my video. That will
sure. Yeah.
Okay. I am on a wired connection, but yeah.
I'll send you a chat if you cut out, right? So just continue, and if something happens, I'll just send you a chat so we don't disturb. Go ahead.
That's excellent. Thank you so much. So I was just going to introduce to you what contaminants of emerging concern are. It's basically an umbrella term that we use to refer to a wide variety of contaminants such as pharmaceuticals and personal care products that are continuously used and introduced into our wastewater, and they tend to be pseudo-persistent. Of particular concern are those that actually induce a biological response or cause some unintentional response in non-target organisms. And I'm thinking of antibiotics or pesticides that are designed to have some kind of endocrine disrupting activity in organisms. And so the treatment of these contaminants is really critical if we are thinking of water reuse, of wastewater. So the extent of treatment for reusing wastewater that we can provide really depends on the end use of that recycled water. So if you are thinking of, just using that recycled water for flushing or for, then it has very minimal treatment and it's called gray water. And on the other extreme, we have potable water for which we have a lot of advanced treatment. And in all of these treatment processes, photodegradation hello?
We can hear you.
Okay, so can you see my screen?
Yes.
Yes.
Okay, thank you so much. So in all of these processes, photodegradation plays an important role as part of the treatment design. So if you're thinking of natural or semi-engineered systems like lagoons or oxidation ditches, then sunlight has a small component of UV light, which actually can cause contaminants to degrade. Or if you're thinking of treatment systems, then we have engineered systems which have UV light for disinfection or advanced oxidation that can cause contaminants to degrade. And so in this talk, I'm going to talk about two parts. So I'm going to speak about reuse of domestic wastewater in potable reuse, and I'm going to talk about heavily engineered systems for these processes. And then I'm also going to talk about the other end when we are looking at reusing agricultural wastewater in natural systems. And so the first part of this work is something that I'm currently working on. This work is done in University of California, Riverside, in the lab of Hai Julio with graduate students Sam Patton and Liang Wu. And I want to really quickly show you two maps of the western half of United States to really stress on why this is important. So this first map here is the precipitation map. And anything that is in yellow or darker red is an area that receives very low precipitation. And then we have a population density map on the right. And if you look at Southern California, we have these twin problems of low precipitation combined with high population. And that has really made this region very water stressed. And so the idea of using wastewater actually stems from this very linear journey that water sees in urban regions. So you have water coming from its source, be it groundwater or any reservoir, and it goes through the distribution systems and wastewater treatment plant, and then wastewater effluent is actually introduced into the ocean. And so this journey is very linear. And so the idea is then to take away the wastewater effluent and treat it and reuse it. But we introduce a lot of contaminants of emerging concern in the distribution system and in our residential use. And so we need to have advanced treatment processes if we want to get rid of these contaminants of emerging concern before any type of potable reuse. And so this work was done in collaboration with Orange County Water District, and it's the world's largest potable water reuse facility. It uses a full advanced treatment system. And what that means is it uses a combination of membrane and oxidation systems for making wastewater effluent potable. potable water. And so the way it works is the facility receives wastewater effluent from Orange County Sanitation District. And it goes through a microfiltration unit, a reverse osmosis unit, and ultimately an advanced oxidation system. And so any larger molecule is then not bypassing the reverse osmosis system and is being collected in the RO concentrate, whereas some small neutral molecules can get through the RO system. And so advanced oxidation is really the last barrier that is there to prevent these contaminants of emerging concern from entering potable water. And so some examples of contaminants that we are looking at are some industrial solvents like acetone, trichloroethylene, and 1,4-dioxane, or pharmaceuticals and personal care products like caffeine, atenolol, and DEET. And so the goal of this project was to study the degradation of these indicator compounds in the UV hydrogen peroxide advanced oxidation process in Orange County Water District. And we primarily wanted to study the impact of reverse osmosis permeate qualities, specifically chloramine. So the question is, where does chloramine come from in the system? So chloramines are biocides that are introduced in this treatment chain prior to the membrane units. And being small and neutrally charged, these monochloramines can make it through the RO, and so they also enter the UV advanced oxidation process. So that's what we want to take into account when we design our UV hydrogen peroxide system. And so to do this, we had an on-site pilot scale reactor. And so we use fresh RO permeate from the facility to build our kinetic model that will describe the treatment efficiency of the system. And we are so able to adjust how much oxidant and chloramines we want to add, and we are able to control those parameters in our pilot scale reactor. So really quickly, I want to just dip into some chemistry here. I won't spend too long, but I hope you all stay with me on this journey here. So what happens when the monochloramine actually goes into the UV reactor? So the monochloramine absorbs UV light and it photolyzes to form chlorine atom and amino radical. The amino radical is not really very reactive with organic contaminants, but the chlorine radical, chlorine atom can then react with chloride to form another reactive chlorine species. And so these two, chlorine dimer and chlorine atom, are really what can degrade some of the trace organic contaminants. And these reactive chlorine species can also undergo a bunch of other reactions to form hydroxyl radical. And hydroxyl radical is what we really want for degrading these trace organic contaminants. And so when you have hydrogen peroxide also in the system, what happens is that you can form more and more hydroxyl radicals that will have greater treatment of these trace organic contaminants that are entering through the RO. So now I want to show you some of the results that we got. So in this graph here, we have the 6 organic contaminants, and on the x-axis is the hydrogen peroxide dose that we are adding for a fixed monochloramine concentration. And what we are seeing is that as more and more hydrogen peroxide is added to this system, you have greater and greater degradation of the compounds that we are looking at, the compounds that are added. So the other thing that we see is that we actually have a combination. So the blue bars here represent the contribution of hydroxyl radicals in degrading these compounds. And the red bar is the contribution of reactive chlorine species that is degrading the compounds. So the other thing that we really notice here is that it's not just hydroxyl radicals that are degrading these indicator contaminants. It's a combination of two. And the dose combination of hydrogen peroxide and monochloramine is really deciding what that distribution looks like. So we also tested the opposite condition where we had a fixed amount of hydrogen peroxide and we increased the amount of chloramine in the system. So when that happens, we're actually seeing the opposite thing. So the more chloramine we add into the system, the lesser and lesser degradation we have. And the reason we are seeing this because the monochloramine actually reacts with the hydroxyl radicals that are being generated. So it's self-scavenging. And so we are seeing this loss in degradation efficiency. So as the monochloramine increases, the steady state of important radicals also decreases. And that is what we saw when we did additional tests with indicator probe compounds. So in this graph here, we are seeing the steady state of hydroxyl radicals that are generated in this system. And you can see that same trend that hydroxyl radical concentration is decreasing as the monochloramine concentration is increasing. And this, of course, is very experimental data. And we wanted to go a step further and see if there's a way we can predict the contamination degradation for any other indicator compound that we didn't test. And so what we did is we took all the 117 chemical reactions that I showed from the schematic a few slides ago, and then we combined that with the data from our bench scale and pilot scale reactor, and we developed this kinetic model. And so now this kinetic model, we are able to detect and predict the degradation of compounds. So we have two models here. One model is predicting the degradation of compounds with hydroxyl radical and chlorine atom, and the other one is with hydroxyl radical and chlorine dimer. So the way to read these graphs is the heat map in the back is our prediction model. And these circles here on the overlaying circles is actual experimental data. And what we see is that this is in pretty much good agreement with our model. If anything, our model slightly underpredicts the degradation efficiency of our system. And so that's actually a good thing because then we are developing this conservative model. So conclusions for the first part, the chloramines definitely scavenge the reactive species that are generated in the UV hydrogen peroxide system, decreasing the treatment efficiency. But then chloramines can also contribute to the degradation of contaminants that are sensitive to reactive chlorine species. And then the prediction model that we have developed in this study can give you a conservative estimate of what any hypothetical contaminant will be treated in a full-scale treatment system. And so all of these studies were, of course, in a very clean system. RO permeate is has very low dissolved organic matter, very low carbonate content. And so we are working with an ideal system where we don't have to worry about a lot of water quality parameters that will affect the hydrogen peroxide UV treatment efficiency. And so I want to go on the other extreme here where I'm going to talk about agricultural wastewater. So agricultural wastewater is very high in dissolved organic matter content. And so that was the primary purpose of this study. So this is some of the work that I did as part of my PhD dissertation in University of Maryland, Baltimore County in Lee Blaney's lab. And some of the undergraduate students who worked with me were Jessica, Daniel, and Temi. So why do we really care about agricultural wastewater? So the main contaminant of emerging concern that we are worried about in agricultural waste is antibiotics. So in concentrated animal feeding operations, animals are fed a high dose of antibiotics to prevent the spread of disease and to increase feed efficiency. And so the antibiotics that animals consume are usually excreted without significant change in them. And so the waste manure that comes out of concentrated animal feeding operations has a very high elevated concentration of different types of antibiotics that are used. And so here are some examples of different classes of antibiotics that have been used in the poultry industry. And the reason this is concerning is because the amount of meat that we consume every year has been increasing, and poultry actually now is one of the most consumed meat in the United States. So this graph is data from the USDA, which tells you the per capita availability of meat. And this is this trend is also reflected in antibiotic sales in the United States. Now, 70% of the antibiotics that are sold are actually used in agricultural applications. And in the past 10 years, we've seen a 25% increase in antibiotic consumption in agriculture. So all of that animal manure waste is usually treated, if it's solid, it is barely treated and it is introduced into the environment, or if it's liquid, it's treated in systems such as lagoon treatment or oxidation ditches. And in these ditches, you have exposure to sunlight, which can cause photodegradation of antibiotics. And that is what we really want to explore over here. So the wavelength range we are now considering is the 310 to 410 nanometer wavelength range, which is more relevant to solar radiation. And so we picked 4 antibiotics that are relevant in this system: ciprofloxacin, chlorotetracycline, roxarsone, and sulfamethoxazole. And the primary goal was to actually study the impact of agricultural dissolved organic matter on these antibiotics. So the way our experiment runs was, we have a tiny UV reactor in which we put the appropriate UV lamps. And then as the antibiotics undergo degradation, we take samples out and then we evaluate them in an LC-MS system. And so this is some of the data for just direct photolysis of these antibiotics. So that's without any dissolved organic matter in them. And so on the y-axis over here is the natural log of the normalized concentration for these contaminants. And on the y-axis is time, or the UV dose or fluence that is provided to the system. And so what I want to highlight here is the wide range over which these antibiotics can undergo degradation. So you have some very sensitive compounds like ciprofloxacin in red, and we have some very recalcitrant compounds like roxarsone. And so we tried to cover a wide range of antibiotics in our study. So to study dissolved organic matter from agricultural systems, we took poultry litter from actual poultry farms, and we used them as dissolved organic matter sources. And then we also used Suwannee River natural organic matter, which is used as a standard or a surrogate DOM in environmental studies. And so we performed aqueous extracts on these DOM sources and then spiked them into our solutions at different dilutions. And then we did the same UV radiation process. And we studied the antibiotic degradation in different concentrations of dissolved organic matter. So I want to jump into a little bit of photochemistry here for you all. So when you have an antibiotic, it can absorb UV light and it can undergo degradation. So that's direct photolysis, what I already showed you. When you have dissolved organic matter present, this dissolved organic matter can absorb UV light and create some intermediate reactive species such as hydroxyl radical or singlet oxygen or triplet DOM. And these can then react with the antibiotic and further enhance the degradation of these compounds. The other thing that can happen when you have dissolved organic matter is that you can, these can screen or these can screen the light that is shining into the system. And so it can prevent the effective amount of photons that are entering. And so that is called screening. And the last way dissolved organic matter can negatively affect the photodegradation is that dissolved organic matter itself can react with some of the reactive species that are present in this system. And so you have some scavenging from dissolved organic matter. So you have this combination of degradation, enhanced degradation, and then screening and scavenging. And so we have some complex behavior from our antibiotic degradation. So we try to capture these in a model. And so the model actually has a component to account for the direct photolysis and reaction with singlet oxygen and triplet DOM. So in the following graph, in the following slide, the graphs that I show you, each of these points here is the observed rate constant, not the degradation, the observed rate constant for degradation. And the line that is running through these points is our model that we developed. And so the next slide has a huge amount of data. So I'll walk you through it. So for ciprofloxacin, we saw that no matter what the organic matter source was, we saw the same kind of trend, that the increase in dissolved organic matter continued to decrease the observed degradation rate. So screening and scavenging really inhibited the treatment efficiency of this system for ciprofloxacin. But when it comes to roxarsone and chlorotetracycline, we actually saw an increase in degradation efficiency. And so we see almost consistent trends no matter what the DOM source was. And then finally, we have sulfamethoxazole, which actually showed sensitization for one type of organic matter source, which is agricultural source. But we saw inhibition only for the Suwannee River natural organic matter. And so the main point I want to make on this very complicated slide is that the source of dissolved organic matter really drives what kind of inhibition or sensitization effect we can expect in photodegradation systems. And so most studies usually use Suwannee River natural organic matter, but for agricultural studies, we want to go a step ahead and make sure that we are capturing all the impact of that agricultural organic matter. And so the conclusions for this part was that the antibiotics in natural systems have a wide range over which the antibiotics can degrade, and the enhancement or sensitization that we see in presence of DOM was really dependent on the kind of antibiotic and the source of dissolved organic matter that we have. So that brings me to the end of my presentation. The overall big picture for this presentation, I hope, gave you a flavor or a taste of what UV-based treatment systems can look like in water reuse systems. We generated fundamental understanding of environmentally relevant photolytic fate in agricultural systems, and then we generated a model that can predict advanced oxidation treatment efficiency for potable water reuse. There have been other projects that I have also dabbled in as I have been through my doctoral and postdoctoral journey, including studying environmental occurrence of CECs, investigating dissolved organic matter characteristics, or even phosphate recovery and reverse osmosis concentrates. But those are all talks for a different day. But I'd be very happy to talk to you all if you're interested in knowing more about some of these projects. And I'd like to acknowledge everybody who has contributed to this work, including the funding agencies and all our collaborators. And with that, I'll take any questions.
Thank you so much, Kiran. I think that we have time to maybe one or two very quick questions. So this was an excellent presentation. If you have a question, please unmute yourself and ask it. And yes, yes.
I have a quick question. If oh, there is somebody that wants a question.
Please,
Utsav, go ahead.
All right, thank you. Kiran, that was really great, showing both of your postdoctoral and PhD work. So my question goes back to your initial work, and when you highlighted that, some of these small molecules actually pass through the aeromembrane, and so some of the contaminants that you worked with, caffeine, I guess dioxane. So are these contaminants able to make through the aeromembrane, and they are present in the before it's actually, if you can go back to that map. In that advanced oxidation process. So these contaminants are already found at that stage.
So I can explain. So these contaminants were often used for studying the treatment efficiency of reverse osmosis membranes, and many of them being small and neutral were selected for this study to evaluate the UV AOP performance. So 1,4-dioxane trichloroethylene, acetone, and DEET have definitely been seen in RO permeate from observed studies. And then atenolol and caffeine were not observed in our study, but then they have been reported in literature.
Okay, that makes sense. So you're using these compounds. I'm sure there might be other compounds that might be detected, but for your study, you limit it to like 4 or 5 of these like model compounds, I guess.
Yes, we wanted to capture a bunch of different chemistries and sizes, and so these were what we went with.
Okay, makes sense. Yeah, thank you.
Thank you.
Thank you. Please just introduce yourself when you're asking questions, so we know who you are. And thank you so much. I think that it's already 12:30, so we don't have a lot of time for more questions. But if you stay until the end, you can ask the questions that you have after the second talk, or you can ask them in the chat. So with that, I think that Manish, are you going to introduce Elise?
Yeah, so Elise, you can share your screen, and let me go ahead and talk about Elise. So Elise is a postdoctoral fellow at AWAG right now. She is one of their first independent postdocs, so it's great to see that these things exist. And I heard her talk or do a workshop at the Biofilms conference, and you'll be pleased to hear her work. It's amazing. She did her bachelor's at the University of Notre Dame, and then she did her PhD with Kyle Beebe at the University of Pittsburgh. And since then, she's been at AWAG. Her postdoctoral fellowship is called the Unique Postdoctoral Fellowship, and she's won many awards. She's been in the MIT Rising Stars workshop. She was a NSF grad fellowship, and she had a pre-doc fellowship from the University of Pittsburgh. So we are very excited to have her here and to hear her talk. Welcome, Elise.
Great, thank you. So my talk today is actually talking about how we can engineer the microbiome of the built environment, specifically using bacteriophages. And before I start, I just want to thank the AEESP AJAR program for inviting me to give this talk and have the opportunity to talk with all of you today on this Friday. My first point of business was actually to introduce myself, but you already did a great job of doing that. So I will just do a shameless plug of follow me on Twitter if you care to see what I'm working on or what I have to say about academia or other things. I know that the audience today is quite broad. Environmental engineering is broad in and of itself. So I just want to give some background information so that hopefully all of you can follow along with what I'm doing. So my work focuses a lot on bacteriophages or phages. And bacteriophages are just viruses that specifically infect bacteria. A lot of them have this kind of cool little rocket shape structure. They're smaller than bacteria, but much more abundant than bacteria. This is an example of some phages that I isolated from wastewater. The turbid area is where the bacteria are growing, and then these clear zones are actually where the phage are infecting and killing the bacteria, creating this clear zone. So phages were actually discovered in the early 1900s, but soon after phages were discovered, antibiotics were also discovered, and antibiotics were sort of seen as this cure-all for bacterial infection. But with the rise of antibiotic resistance, and as well sort of this shift in focus from thinking that all bacteria are bad to that actually some bacteria are quite good or neutral, there's been a lot of renewed interest in using bacteriophages. Bacteriophages have a lot of different advantages. You can actually target specific bacterial pathogens with phages. They're harmless to human cells. They can't infect human cells. And they can actually resensitize antibiotic-resistant bacteria to antibiotics, which is a really big pro with using bacteriophages. And they're relatively easy to isolate. But there's also some challenges with working with bacteriophages. One of them is that compared to a lot of other microbes, they're very understudied. We just need to know more about them. They can be too specific to their bacterial host, so you might actually need multiple phages in order to clear a bacterial infection. They're also difficult to regulate, and we don't know their potential environmental impact if we were to release a lot of phages into the environment. As well, I just want to introduce the concept of opportunistic pathogens, and specifically Pseudomonas aeruginosa, because this is the bacteria model that I use for my experiments. So opportunistic pathogens are pathogens that are normal inhabitants of the water environment, either in nature or in the premise plumbing system or the built environment. And typically they're not harmful unless they come into contact with more susceptible populations. So people who are older or immunocompromised. And Pseudomonas aeruginosa is particularly important because it has a lot of antibiotic resistance and is implicated in a lot of hospital-acquired infections. So it's actually one of these escape pathogens, which maybe you have heard about, and as well as a WHO priority pathogen. So I want to motivate my research by walking through a little bit of an example here. So say we have a shower head, and in the premise plumbing system, maybe we have some Pseudomonas aeruginosa. If we have an individual who has cystic fibrosis that's using the shower, they might actually be susceptible to getting an infection of Pseudomonas, and maybe they get so sick that they have to go to the hospital. Now, in the hospital environment, there can be many different sources of bacteria. One, of course, being the patient himself. This patient could then cough or move around the room and create foments that are then spread across surfaces. And as well, something that we're very concerned about is actually how we treat the patient. A lot of these soft, flexible plastics, such as catheters or IVs or tracheal tubes, can actually be very prone to having biofilms build up on them. And all of these scenarios are areas in which we could have potential phage application to eliminate these pathogens. So for instance, a lot of current phage work is actually going into treating the patient themselves, the actual disease, with phages. As well, we could think of sort of a disinfectant type phage-based product to eliminate these pathogens on the surface. And as well, there's been a lot of research into looking at how we can treat biofilms in these soft, flexible plastics, or even pre-treat them so that the biofilms can't form in the first place. But luckily for our patient, let's say they recover and they're able to be moved out of this room. So right away, some of the sources of bacteria will be eliminated. But we still have these lingering fomites that can be around the room. So currently the best practice is with chemical disinfection for room turnover. And we know that a lot of these bacteria can actually build up resistance to these chemical disinfectants. So we might be able to eliminate some of them, but they could regrow or just be resistant. As well, bacteria living in biofilms are as well particularly resistant to chemical disinfection. And then say we have another patient that is brought into this room afterwards, and they could either be walking around the room or have a healthcare worker that's interacting with the surfaces around them and then interacting with the patient. And this bacteria can be reintroduced into this new patient. And now this becomes a hospital-acquired infection. And these hospital-acquired infections are actually a big deal. Pseudomonas aeruginosa alone was responsible for about 51,000 of these in the US yearly, and 13% of these are multi-drug resistant. And hospital-acquired infections in general are increasingly multi-drug resistant. So in high-income countries, the rate of patients that go into a hospital and acquire a secondary bacterial infection is about 7%. And this is actually higher in low-income countries at about 16%. And these secondary infections greatly affect patient outcome. So just as sort of a current example, it's thought that 1 in 7 COVID-19 patients develop a secondary bacterial infection while they're in the hospital. And as many as 50% of the deaths from COVID have an additional hospital-acquired infection. So like I said, this can really impact the patient outcome. So looking at all of this just sort of motivates my research and what I'm interested in. So instead of treating the patient or treating the disease, I'm actually looking at how could we use bacteriophages in order to prevent the disease from happening in the first place. So specifically, I'm looking at, can we combine phages with chemical disinfection? There's been a lot of research into synergisms between bacteriophages and antibiotics, as that's how they're often applied in humans. But there's really not a lot out there about how bacteriophages could interact with chemical disinfection in this sort of scenario. So looking specifically at my research questions, the first one is just in general, can combining these two types of treatment effectively eliminate surface-associated Pseudomonas aeruginosa more effectively than either treatment alone? And then another question that I wanted to know was, does the order of these treatments matter? It's really important to note here that in my research, I applied these treatments sequentially. So first applying phage, then the phage was removed, then applying chemical disinfection, or in the alternate scenario, first applying chemical disinfection, that disinfectant removed, quenched, and then applying to phages, so that we can sort of decouple this. But on the other hand of that, my last question is actually, what is the effect of these disinfectants on the phages themselves? And there's sort of a couple of reasons why we might like to know this. First of all, I think something that we should really be concerned about is actually, if we do have these released into the environment, could that be a problem? And we probably don't want to do that. So actually, maybe if we treat with phage in the built environment, maybe we do want to follow that up with chemical disinfection to inactivate the phage particles themselves. And then as well, this could also be important to know in case we did want to have a formulation where we apply these simultaneously. So in that case, it would be important to know that the phage is not inactivated by the disinfectant. So I have 3 experimental setups that I tested for this. The first one here being spot inoculations. This involves a plastic cover slip that's laid flat into a well of a 12 well plate. The Pseudomonas aeruginosa was spotted onto the surface here. It was allowed to dry just so that there's no remaining liquid, and then it was proceeded into treatment. Then we have our dry biofilm model, which is here. In this case, we have a plastic cover slip that's inserted perpendicularly into the well. And we get a biofilm that forms at the air-water interface here. We can then remove this plastic cover slip, and we dried it for 2 hours before proceeding to treatment. And then our third scenario is the wet biofilm model, which you can see here. This was done in 96 well plates. We put our media in. Again, we get the biofilm that forms at the air-water interface, and then this is treated. So firstly, I want to talk about the spot inoculations. So for all scenarios, I treated with 2 different types of phage, not at the same time, but one or the other. Phage JG4, which was gotten just from a strain collection here in Europe, and phage P1, which I isolated from wastewater here in Zurich. This phage treatment would then be followed by sodium hypochlorite or benzalkonium chloride treatment. And I'm not going to highlight the benzalkonium chloride work just for the sake of time. After treatment, the cells were recovered and then quantified via flow cytometry for total cell count and intact cell counts. And I'm only going to show the intact cell counts today. again, just in the interest of time. So some results. So firstly, I just want to thank my master's student, Anina Cole. She did this particular part of the research. So these graphs, what you can see is on the x-axis, you have increasing concentrations of either sodium hypochlorite or of the different phages. And then on the y-axis, you have the intact cell count that's given as a log 10 reduction. So if we just look at the sodium hypochlorite, as we increase concentration, we also increase our log removal. And then as well, when we look at our two phages, as we increase phage concentration, we as well reach total inactivation as seen by the limit of quantification of our flow cytometry method. So essentially, we would have liked to do combination experiments in this model, but our phage are so effective at inactivating the bacteria that at least the method we used, being the flow cytometry, we wouldn't be able to see any sort of synergy because we would have to use such a low phage dose that it wouldn't really be effective. But this is really good to see, first of all, that these phages are able to inactivate this bacteria very quickly and easily. So next, going to our dry biofilm model. Everything's the same. We treated with the same two phages, followed by sodium hypochlorite treatment, or in theory, followed by sodium hypochlorite treatment, and again, quantified on FCM. But here we see something a little bit different. Again, as we see increasing sodium hypochlorite, we maybe see a bit of inactivation, but it's certainly not as pronounced, likely due to it being a biofilm instead of just free bacteria on the surface. And as well, when we look at our phages, we essentially see no log reduction, which is certainly not the result we were hoping for. But we sort of thought about it and hypothesized that since phage need actively growing host in order to infect and replicate, that maybe this drying process really had a significant effect and impaired this treatment. So how we tested this was we took some of our dry biofilms and we dried some of them like normal for 2 hours, and then some we recovered after not drying at all, just after overnight incubation. And we did growth curves with these starting communities. And as you can see here, there is no significant difference in the growth rate or the carrying capacity of these two communities, but where they do significantly differ is the lag time. So for the non-drying cohort, the lag time is about 3 hours, and for the dried ones, it's about 7 hours. And what's important to note is that we treated with our phages for 4 hours. So when we think about this, it actually makes a lot of sense that maybe we didn't see so much inactivation in our dry biofilms. I think if we increase this treatment time to say 8 hours or even more, maybe we would see this inactivation. And then our last model that we looked at are the wet biofilms. So the difference in this model is that we changed the order of the application. So we looked at first treating with phage, and then followed by chemical disinfection. And then as well, we did test where we first applied the chemical disinfectant and then the phage. And again, the chlorine was quenched before the addition of the phages so that they wouldn't inactivate the phage themselves. So this is some of our data here. On the left-hand side, you're seeing the model where we treated with phage first, and then followed by the chlorine. And then on the right, you're seeing the model where we treated with chlorine first, followed by the phage. So on the x-axis, again, we're seeing increase in phage concentration, and on the y-axis, we're seeing the log reduction value of the intact cell count. And the different colors correspond to different chlorine concentrations. So in general, this makes a lot of sense. As we increase our phage concentration, we see more removal. And as well as we increase our chlorine concentration, we as well see more removal. And then this is seen to a lesser extent in the chlorine first model. But in order to sort of get a real idea of, is there an added benefit of combining these treatments? This is how I chose to represent that. So what you're seeing here is sort of a grid that is showing you which of the treatments might have dominated for each of these combinations. So essentially, if you're seeing a blue color, that means that the chlorine treatment dominated. If you're seeing a yellow color, it means that the phage treatment dominated. And if you're seeing a green color, it means that that result was significantly different from either the phage alone, from both actually of the phage alone treatment or the chlorine alone treatment. So we are seeing some sort of added benefit for these green ones. It might not necessarily be additive, but there is some benefit of combining these. And as well, this makes a lot of sense. At very low phage concentrations, we see that our chlorine dominates the treatment. At low chlorine concentration, we see that the phage is dominating the treatment. And then at sort of these higher concentrations of both, we see that there's some synergy. For the chlorine first treatment, this is not as pronounced, but this is actually easier to see with our other phage. So this is our phage P1, and as we can see here, there's a lot of blue. So at these higher chlorine concentrations of 200 or 20 ppm, basically the chlorine regime dominates entirely the inactivation. We do get some added benefit of our phage at high concentrations for a very low chlorine dose, but it's actually even above just the phage alone. And so basically my hypothesis for this is that we're stressing the cells when we're applying this chlorine first, and so that then by the time that you're adding the phage can't effectively attach or infect their bacterial host, and that this is hurting sort of the treatment of the biofilms. And then lastly, I just want to touch on looking at the phage inactivation themselves by the disinfectants. So we have our two phages here and two disinfectants, sodium hypochlorite, which is this Cl, and benzalkonium chloride, which is this BAC. So for the sodium hypochlorite, both phages are actually inactivated quite quickly, reaching over 7 log of removal after just 15 or 50 CT value units. And for benzalkonium chloride, they are more resistant. So phage P1 is actually not inactivated at all until it reaches this threshold, and then it follows some first order decay. And then phage JG4 actually does drop a little bit when you reach 20, but then it stays constant. So even with this, or when you look into the literature, we can see very broadly that this depends a lot on the phage. And so any sort of formulation you make, you'd have to take that into account. So coming back to my research questions, can combining these two different treatment types be more effective than either of the treatment types alone? We do see that phage and disinfectant used together can improve removal. Does the order of the treatments matter? It does seem that phage treatment followed by disinfection is more effective than the reverse. And how do the disinfectants themselves affect the phages? So our phages are susceptible at least to sodium hypochlorite, but there's a wide variability between disinfectant types and phage types as well. So could we use the disinfectant to doubly eliminate the active phage particles? Maybe. It seems more likely in the case of sodium hypochlorite in our scenario. Or could we use both of these applied together? Maybe. Again, for us, this seems more likely with the benzalkonium chloride model, for instance. But this would have to be evaluated for each particular formulation. So moving forward, sort of my whole research path is sort of focused on this interplay of phage ecology and how this is related to the built environment and human health. So I want to do more research in control of pathogens, which is mostly what I was showing you today. There's much more research that can go into this area. As well, a big vein of my research trajectory is looking at potential unintended consequences of phage treatment. So looking at the environmental impact. And then another third, equally important sort of research area that boosts these other two is how we could potentially be using phage to monitor water quality. Hopefully I'm able to do a bit more research before my postdoc here at AJAR ends. But as all of us know, 2020 gave us some other ideas. So actually a lot of the past year I've been helping with COVID-related research and was not able to do as much of my own research, either due to restrictions of lab access or other things. So we'll see how it goes as we continue in this pandemic. And then lastly, I just want to thank some people. So my department here, the Environmental Microbiology Department, for funding my research, as well my specific group, Pathogens and Human Health, which is headed by Tim Julian, and as well my master's students who have worked with me, Anina Kuhl and Anne Vescovi. And with that, I'd be happy to take any questions that you might have.
Okay, you can raise your hand and I'll call on you, or you can introduce yourself before asking questions, but let's first thank Elise. Yeah, so tell us where you're from and something about yourself before you ask your questions. So I have a question while people warm up. With these phages, why do quaternary ammonium type disinfectants work? Do they have a lipid envelope on some of these phages, or do they work even with just the other kind?
Yes, so this is a very good question. Actually, the work of my other master's student is actually looking into this a little bit. She had to abandon her lab-based research, of course, because of coronavirus. So actually, her master's thesis turned into sort of a literature review, collecting data on viral inactivation due to quaternary ammonium compounds, and sort of looking at the viral characteristics and what may play into this inactivation. And something we found very strongly was that having a membrane greatly impacts how these are inactivated. So enveloped viruses are able to be inactivated way better than non-enveloped viruses. I don't know particularly the phages that I'm working on, what their structure is. But it's reasonable to think that these are non-enveloped phages and therefore could be a little bit more resistant to the quaternary ammonium compounds.
All right, Lisa, do you want to ask your question?
Hi, great presentation. My name is Lisa and I'm from the University of Pittsburgh. I was just wondering, can bacteria evolve resistance to these bacteriophages eventually?
Yes, absolutely they can. Sorry, did you have a second part to your question? Did I cut you off there?
Okay, so I guess like what's the mechanism of how they interact with the bacteria? And so would that mechanism of resistance be directly correlated to that?
Directly correlated to what exactly? Sorry.
So yeah, I guess I'm not familiar with phages. So like how do they work to inactivate bacteria, and what's the defenses that could potentially be gained by the bacteria to evolve resistance?
Yeah, great question. So bacteria absolutely can become resistant to bacteriophages, just like they can become resistant to chemical disinfectants or antibiotics. This is mostly thought to be achieved by altering the surface structure that the phage attaches to. So an important part of phage infection is that they have to attach to their bacterial host by some sort of cell receptor on the outside of the cell. And this then allows them to infect their DNA into the cell, etc. So when these surface structures are mutated and then the phage can't attach anymore, we've actually seen a lot of evidence that this then allows the bacteria to be resensitized to antibiotics if they're resistant to antibiotics. because as well, a lot of these surface structures play a key role in how the bacteria are resistant to the antibiotics as well. Does that answer your question?
Yes, thank you.
Yeah, I'm going to call on Holly next. Go ahead, Holly.
Hi, can you hear me?
Yes.
Yes.
Okay, great. I'm Holly Adedrumo. I'm a PhD student at the University of Michigan. And yeah, I know Kieran, and as well, I was at undergrad at UMBC. So my question is regarding the bacteriophage's specificity. So you mentioned earlier that bacteriophages infect specific hosts. So are the bacteriophages that you're working with naturally occurring, or is there potential to make them more specific to different strains or different, like as the bacteria evolve, is there ways to make it more specific to those other strains?
Yeah, so great question. You bring up a couple of important points. So again, sort of looking at how bacteria gain resistance, there's also actually been a lot of evidence that if you treat with multiple phages at once, these will have different surface receptors, and that then you're much less likely to get these bacteria escaping that are resistant, because maybe they'll gain resistance to one of the phages, but not the other one, for instance. And then as well, I had another point if they're naturally occurring. So phages are, sort of the theory is that phages can be found wherever their bacterial host is found. So for instance, my phage P1 that I use in my experiments here, I isolated from wastewater. Wastewater is usually a great resource to find bacteriophages. And then essentially you just seed in the bacteria that you want to find phages for, and then that's how you can isolate them. But as well, like if you're looking specifically in a patient, you could like sample their wound, for instance, if it's like a surface infection, and you can likely find phages there. Or as well, there's a lot of bioengineering applications in this. So I would say it's actually more, there's more being done to actually expand the phage host range than to limit the phage host range. Like phages are very specific. They can even be specific to the strain, not even necessarily the species, but to the strain of the bacteria. And so there's actually people who are looking into how can we change the tail fibers so that these phages could attach to a broader range of bacterial hosts.
Thank you.
Okay, I'm gonna do one more question and then we can be more informal. Tian Ki, I don't know if I'm saying this right. Can you go ahead and unmute yourself?
Yeah, thank you. Hi, Elise. A very nice and interesting talk. And I'm Tianqi from current postdoc at Georgia Tech. And my question is, when you design the experiment for the sequential application of phage and disinfectant, how do you design the time for each part of treatment?
Yeah. Great question. So for this, it was mostly looking into the literature to see what other people are doing. For a lot of chemical disinfectants, the recommended contact time is 10 minutes. So that's what I used for my contact time for the chemical disinfectants. And then as well, I did do some preliminary experiments looking at how long I might want to treat with phages for. Some people treat for phage for 24 hours, but the problem with doing this is that then you can see regrowth due to phage resistant populations. And while that's very important, it wasn't really the question I was trying to look at specifically with this study. So I landed on 4 hours because it's enough to see this initial inactivation, but not long enough where you're then seeing the regrowth of these phage-resistant populations.
I see. Yeah, thank you. Yeah, I wonder, like when you change the time for each treatment process, and will you get a different output? For instance, the disinfectant, if you apply for a longer time, and they probably can have a larger contribution, such as like that. Yeah.
Yes, potentially.
But yeah, thank you.
Great. Let's thank Elise one more time, and then I'll ask her to stop sharing. Yeah, and now it's open season. I'm going to stop recording.